Welcome to the ATiO Podcast, the ultimate gateway to first-hand insights from world-renowned experts in the realm of advanced therapies and orthopaedics, unraveling the latest advancements and perspectives. Join us as we navigate this intricate landscape right here on the ATiO Podcast.
We are honored to welcome Professor Dr. Ivan Martin today, a true pioneer in the field of tissue engineering. Professor Martin is currently the head of the Tissue Engineering Research Group at the University of Basel, where his work focuses on developing innovative approaches to cartilage regeneration and repair. Professor Martin has an impressive background with over 300 peer-reviewed publications and decades of experience in the field. He's widely recognized for his contributions to cartilage regeneration, particularly his development of the nose-to-knee technique, which involves using nasal cartilage to repair damaged knee joints. His research has the potential to revolutionize how we treat cartilage injuries, moving us closer to more effective and personalized therapies. In today's episode, we will dive into Professor Martin's journey, the exciting projects coming out of his lab, the challenges and opportunities facing the future of regenerative medicine. We'll also touch on the clinical potential of tissue-engineered products and what the next decade could hold for the field. Pleasure to welcome you, Ivan. I really loved your presentation with the title From Nose to Knee at our latest conference. You're really doing incredible work in the field. Let's start with a brief introduction of yourself for the audience.
Yes, so I'm by background actually an electrical engineer, and I have worked in this area up to my master thesis, trying to implement neural network principles to calibrate robot systems in space, so very far away from tissue engineering and regenerative medicine. And in the context of this neural network programming, I had to follow a course at the university about natural biological neural networks. And this was a killer for me. I was fascinated by the complexity of biology, by the possibility of discovery. And then I decided to start a PhD in biological sciences, still with some connection with engineering. And this has brought me somewhat by opportunity also, thanks to the options which were open at that time for a stupid electrical engineer in the field in my university. And I was lucky also to enter this area in the period of the hype of tissue engineering. It was the early 90s. In 1993, you may remember, Bob Langer and Jay Vacanti published the first paper with the title Tissue Engineering. And for me, it was absolutely a thrill to think that I could work in research with a possibility to develop new therapies that could revolutionize the medical field. And this for me was the path. And later on, I was trained in biomedical engineering at the MIT Harvard Health Science and Technology Division. And still a few years back, I was trying to combine these biological and engineering perspectives into clinical translation. And today I find myself still in this path, but also with a large duty in management of a big department of biomedicine at the University of Basel, so that my skills progressively evolved from electrical engineering to biology to biomedical sciences. Now, I must say, unfortunately, predominantly into management of science.
That's a really inspiring biography. Can you share some of the exciting projects your lab is currently working on?
So if I have to mention just a couple of projects, I would say what you initially referred to, so the possibility of growing engineered patches of cartilage, starting from a small biopsy taken from the nose for the regeneration of articular cartilage in the knee, in the shoulder, now in the ankle joint, in the elbows. This is, for me, where I see the largest interest in my group and also by the scientific community at the moment. So it's the program that we generally call the nose to knee program. I could also mention a direction of research where we are still engineering living tissues out of living cells, but then we devitalize the tissues prior to use them as grafts. And we preserve in this way only the extracellular matrix with all the signaling molecules, the structural proteins. And this, in the context specifically of bone regeneration, is an approach for engineering biomaterials in the end that can be customized in composition, structure, function based on the cells which are used, on the culture conditions, but they are cell-free at the end, and so they could be used as off-the-shelf materials. And I could still mention the engineering of three-dimensional culture models that we are pursuing specifically in the context of modeling the bone marrow niche in order to study interaction between stromal, endothelial components with hematopoietic cells in physiology, in malignancy, in a way to develop human organotypic models where we can discover and develop new strategies of therapies for general hematopoietic pathologies.
A lot of interesting programs and an insane workload. So I suppose you also have a great team standing behind of you.
Absolutely. It is basically my team that pursues mostly all these lines, and I enjoy discussing with them some updates and sketching the design of a new experiment, but it's their duty and their honor to harvest the fruits of these projects.
Beauty of research. As you know, I'm also super interested in cartilage repair. So I would like to talk a little bit about the nose to knee program because you already mentioned the term off the shelf. What would be the final outlook? Can we expect an off the shelf product?
So this is a question that I ask myself and we ask ourselves in the team many times. What do the degenerated or injured joint requires as a component to revert the degeneration and to maintain a physiological function and homeostatic biology? And in my view, in certain advanced cases of degeneration, you do need a living cellular component to regenerate new tissue. And even if this only forms the template for further remodeling thanks to the resident cells, it's difficult to imagine that just by delivering certain signals to the cells at the local site of injury, you will be able to convince them to regenerate the full large defect. But this could be different in cases of smaller defects. Obviously, it could be well different in cases of non-cartilage, but other types of pathologies. But for cartilage repair, I think based on the current knowledge, we are still bound to have to deliver living cells. In cases, I'm referring to the more severe damages and advanced stages of degeneration, which is what we are targeting in our nose to knee program.
And at what point are you currently in the pipeline of this program? Do you already have some in vivo data on that?
So we have treated now, as of today, more than 100 individuals with focal cartilage defects. We have run one phase 1 and one phase 2 study, which indicate the promise of this method and the fact that it is important to have a well-organized functional cartilage tissue, providing an advantage as opposed to simply delivering cells in a gel or in a suspension. Now, we also recognized in these 100 cases that those more critical lesions, because of a revision surgery, because of the size, were also benefiting of this treatment. And so we have started treating as a pilot series some patients with advanced osteoarthritis. And these patients, which were otherwise would have had as a alternative just the implantation of a prosthetic device, are still doing fine a few years after the implantation of this engineered cartilage. And based on this and recognizing that this is the unmet medical need, or if you want, the market need, then we have applied and received funding for treating now 225 new patients with osteoarthritis, and specifically with a patellofemoral osteoarthritis, which is a specific form, which is quite frequent, which is aiming for us at restricting a bit the breadth and heterogeneity of otherwise a very variable patient population. So coming back to your question, where are you now? We are at the beginning of another phase 2 study with for the implantation of engineered patches of nasal cartilage in osteoarthritic patients in 225 individuals, this time with controlled arms in the clinical treatment. So we will be able not only to assess, let's say, feasibility and safety or promising future directions, but effectively to test efficacy against other treatments.
Those are really amazing news, especially the patellofemoral arthritis is something that is really hard to deal with in the clinics. Some detailed questions. What size of focal defects were you treating in the past studies? And did you also try it on different joints?
So the maximum size that we have treated until now is about 8 square centimeters. For the patellofemoral osteoarthritis, these are also called kissing lesions. So we have defects on both sides of the articular cartilage. And so for these patients, we are engineering actually 2 patches of nasal cartilage. And so these will be engineered in the size of 4 by 5 centimeter each. So it will be 20 plus 20 square centimeters of engineered cartilage. Obviously, they will be shaped and cut to the size and geometry that best can fit the defect. But this is the technical limitation maximum of the technique. In the context of a temporary authorization by our regulatory agency, Swissmedic, also treated patients with shoulder defects, ankle cartilage defects, and we are planning now to introduce the technique also for elbow defects. For shoulder, it seems to be working well, although ideally our orthopedic surgeons prompt us and push us to develop osteochondral implants that could better fit their standard surgical procedure. And for ankle cartilage, I must say the early results are not crystal clear. And also there, I have the impression that in most defects, it's not only a problem of the cartilage, it's also a problem of the subchondral bone. And therefore, our procedure may not be ideal as it is now.
Could you explain step by step what you're doing exactly, also for maybe a person that does not have such a profound medical background and starting in the nose?
Yes. So the patient is going in an ambulatory setting for a harvest of a biopsy from the nasal septum, which is 6 millimeter in diameter. And this is taken under local anesthesia, usually not painful. The patient can go home after half an hour, and this defect does not have any functional consequences for breathing or for pain, for anything. So it's a relatively minimally invasive procedure to harvest the piece of nasal cartilage. Out of this piece of cartilage, we isolate cells and we expand them in number. What we reach is tens of millions of cells, which are then, after exactly 2 weeks after the harvest of a biopsy, which are then loaded onto a collagen sponge. And at this stage, the cells receive the signals that they should stop proliferating and they should start generating new cartilage. And this process lasts for another 2 weeks. And at the end of the total of 4 weeks, we have a piece of tissue, effectively, where the biochemical composition and also the mechanical properties approach those of a native. And at this point, the product is released based on certain criteria, which have been agreed with our regulatory authorities, transported to the operating room for implantation after being shaped accordingly.
And could you explain again for the audience why nasal cartilage, or where are the difference between the cartilage we know that is present in the knee and in the nose?
I'm very glad you're asking this question. I was hoping for that. So we have tried over 20 years of research, different possible sources of cells to generate with repeatability across the heterogeneity of many patients with different genetic backgrounds or different ages or different lifestyles. And the only source of cells that enabled us reproducibly to generate this good quality of cartilage in the petri dish was the nasal chondrocytes. And even the so-called stromal stem cells from the bone marrow were not competent to generate this quality of cartilage at this level of reproducibility. And this has pushed us also to try to ask ourselves why, where are the instructions encoded in the nasal chondrocytes to be able to be so potent in regenerating new cartilage. And it seems that these properties derive from the fact that the nasal chondrocytes are, from an embryological development standpoint, are derived from the neural crest, which is where our hierarchically superior organs, the brain, the eyes, develop. And epigenetically, there seems to be an imprinting of a higher plasticity, so capacity to adapt to new environments, and regenerative capacity, which is exactly the two features that we require if we implant these cells and these tissues in a different location where regeneration is required. And this is still the tip of the iceberg of what we hope to discover about where epigenetically, specifically, are these instructions encoded, because maybe this could open new ways of reprogramming other cell types, maybe including cells of the own joints.
Amazing. I'm super curious of the results of your upcoming studies, and I think everybody is. What are some of the advantages of your technique, nose to knee, over classical ACI that is already used in some clinics?
So in the phase 2 study that has completed already, we have exactly compared the engineered nasal cartilage as a graft versus the delivery of cells, the same cells actually, so still from the nasal septum without being pre-cultured on the material, so delivering them in the form of cells and not in the form of tissue. And we had a statistically significant superiority in the clinical outcome for the engineered tissue arm, both in terms of self-assessed scores, so the patients were doing better in terms of return to sport, pain, and life quality, but also from the objective radiological assessments through gadolinium-enhanced MRI. And so we could see that the engineered cartilage versus the implantation of cells which are not pre-cultured leads also to better composition of the tissue up to 2 years after implantation, and to a beneficial effect also in the surrounding area surrounding the defect. Why is that is subject to discussion. So it could be because of the initially superior mechanical properties of the graft, so it could better transduce the loading patterns which are activated even in postoperative rehabilitation. It could be because of the better protection of the cells from the extracellular matrix of the tissue, so they are more resistant to the inflammatory insults of an injured joint. It could be because of the signals which are embedded in the extracellular matrix and that are somewhat healing and rescuing the environment. So this is an interesting open question, but clinically we do see a superiority for engineered cartilage versus delivery of cells.
Summed up, we could say a better and more functional graft, which could revolutionize the current clinical landscape of cartilage treatments.
I was a junior PhD student when the vision of an engineered tissue for cartilage repair was first put forth. And I recognize that in these, unfortunately, already 30 years of time, there has been no proof that engineered cartilage can be superior. And so I do agree with you that this finding can be revolutionizing because it could open new avenues of investigations or rejuvenate avenues of investigation that have been abandoned over time.
Speaking of almost 30 years of experience, what do you consider the most significant breakthrough in tissue engineering in the past decade, and how has it influenced your work?
So, unfortunately, there is not much adoption at a broad scale of engineered tissues in the clinical practice. So, I do not see that this revolution of tissue engineering has yet taken place. But I think there has been in the past decade or so the development and evolution of technical tools which are absolutely game changers in the field of biomedical sciences and therefore also in tissue engineering. If I think of single-cell RNA sequencing, of CRISPR-Cas gene editing techniques, the multiplex imaging, the organs on chip development, all these are rather technical enabling tools, which I think will bring fruits for tissue engineering in the next years.
And how close are we to seeing tissue engineered products being used in routine clinical practice? In your opinion, what are the biggest challenges in achieving this?
So my opinion, disclaimer. So in my view, engineered tissues, in order to be adopted clinically on a routine level, they need to be effective and cost effective. I would say that the cost effectiveness can be somehow generated or developed through the use of scaling up or scaling out technologies, bioreactors, new technical tools. And although this is an important problem for the clinical adoption, so the cost, it's, in my view, a secondary problem. The main problem is to demonstrate effectiveness prior to cost effectiveness. And to demonstrate effectiveness in a reproducible way, we need, first of all, to understand the mechanism of action of the engineered tissue. So what are the biological processes which are activated upon grafting the tissue in the injury for regeneration? Until we have defined this, it will be always somehow of a trial and error. Does it work? Does it not work? In some patients it will, in others it won't. If we instead understand the mechanism of action, then we can define, first of all, in-process controls and release criteria for the graft itself. So which properties does it need to have to be released and used? And second, we can define the patient category that is expected to benefit of this particular graft with these particular qualities. And this combination of release criteria and patient target specification will improve the robustness, the reproducibility of the outcome. And this, in turn, yes, it will address effectiveness, but it will influence cost effectiveness because we will know that we don't need to use an engineered tissue in all the, let's say, 10 patients that I receive in the next month, but only in those 2 which I know will have a benefit. And this is the key for guaranteeing an adoption of a field and also a strengthening of the commercial interest by industry in this type of approaches.
Speaking of different kind of tissues, we already have on the market some skin and bone graft products with promising data, but we're, as we already mentioned, plenty of times are lacking great solutions in cartilage treatments. What are some of the limiting factors when we speak about cartilage?
So you made very good examples. So let's take skin. So for skin, it's clear, for example, in burn people, that you need a component of stem cells with capacity of self-renewal that will, let's say, form a new patch of skin to be implanted, but also that will be able to sustain the continuous development of and let's say remodeling of skin through the next decades of the life of the individual. So we know the mode of action and the biological process that is absolutely required so that the potency can be guaranteed. For bone regeneration, we are not so clear yet we know something more about the capacity of certain grafts to be osteoinductive, for example. So how a graft can recruit a population of progenitor cells locally into becoming osteoblasts and into forming new bone that obviously also will remodel and will be then sustainable, maintain the regeneration which has been induced. For cartilage, I personally don't know what is necessary to activate. So my assumption in the work that I'm doing is that you need an initial population of living cells to regenerate tissue. But what happens to this tissue over time? Will it be remodeled by cells in the joint? And which cells? Will it be certain superficial zone chondrocytes that reprogram themselves? Or will it be synovial cells? Does it require the contribution of skewing the immune properties of a joint through macrophage polarization? All this is not known. And until we understand this, we cannot control these processes. And so we cannot guarantee success in patients.
So there's definitely still a lot of basic research to be done in cartilage.
Absolutely.
How important are partnerships in advancing your research? And can you give an example of a successful collaboration?
So at some point in the clinical translation, it's clear that the academia can only offer a certain development of concepts and proof of principle, maybe up to phase 2 studies. But then in order to move the developments into clinical adoption, you need an industry partner, both because of the amount of resources which are required, but also for distribution, commercialization, connection with the clinicians, all this. So this is the obvious answer. I would like to give also a less obvious part of the answer, which is that industry can help us scientists jointly design studies that have more realistic chances of future implementation in the clinic. So they can bring us down to earth and tell us, look, this is never going to work. No clinician is ever going to buy this. It has to be easier to use, or the surgical approach has to be different. And therefore, I think the most successful cooperations with industry are those which are carried out not with the industry as a sponsor and a scientist or university group as an implementer of a certain concept, but at equal height level, where we effectively collaborate and develop solutions together. And I can mention here a very long-term and successful cooperation that I have had with some of the material developers, companies, Geisslich, specifically for the chondroguide membrane, or Finceramica Farenza for the Angipur scaffolds. And the reason why we have been working very well with these groups is not only because of the availability of materials, but also because of the discussion with them thanks to their competencies, and also having access at some point to the clinical data that they have acquired through the past years, which are extremely relevant for us to plan the new therapeutic strategies.
So you bring a lot of great examples for one of the core concepts of the ATiO interdisciplinary exchange.
And that's why I very much enjoy your meetings so far in Berlin. Of great format, excellent discussions, excellent forum.
Speaking of resources, funds are one of the most limiting factors in research, obviously. So let's imagine unlimited resources. What would be the one project or goal you would most like to pursue?
This is a question that I typically ask my students, trying to get some good ideas. It's always a tough question. If I had to focus on an area of science, clearly it cannot be a single project, but more of a program, I would say I would bet on the field of regenerative immunology, where also, by the way, you have a great school at the Charité in Berlin. So understanding which signals need to be delivered to predispose the site to be repaired to regeneration. I think this is extremely important. There are some excellent data that show this, but so little has been done so far. And in this context, connecting the clinical phenotypes, the molecular endotypes, and the response to therapy of a patient database, which is as large as feasible with unlimited resources, maybe through artificial intelligence tools, will enable us to find associations which maybe are unpredictable in a biased way of analysis, and that could revolutionize the field. So basically understanding which immunological features can and have to be considered and prioritized along with the molecular associated signals to predispose to regeneration.
Yeah, it's definitely a super interesting topic, and there's a lot of work being done at the US Wolf Institute by Dr. Katharina Schmidt-Bleek, who is really at the forefront there, researching on the immunologic part in regeneration.
Very much of a fan of the work that is being done there, yes.
Let's continue with a more personal question. What is something about your work or your persona that might surprise the audience?
I don't know how many people know that on the weekend I have a second job selling ice cream, because my wife, she is a producer in our house, of a successful producer of handmade ice cream with the best possible quality of ingredients and organic when possible, fresh fruits. And she sells this through the week, especially on the weekend. Of course, the demand is higher. She has a lot of employees already, and this happens in the ground floor apartment of our house in front of my vegetable gardens. And there over the weekend, I very much enjoy helping at the selling and serving customers, seeing faces of happy children living with a good satisfaction. For me personally, it's part of a balance, and my activities that I need. It still involves a lot of interactions with people sometimes, and often also I manage to meet colleagues in a more informal environment and still discussing something about work. But also it follows the same principle that I try in this moment to deliver the top quality that can be generated. So yeah, this is probably the unknown part of my work.
Amazing. Definitely a surprise to me. And I imagine it's a beautiful way to relax on the weekends and also get access to free ice cream, right? Do you also use your own fruits? Because you mentioned your vegetable gardens.
Yes. My wife last week made a nice sorbet of red and black ribes, Johannisbeeren, and the cassis, along with the figs of our tree.
Wow.
A very special creation.
So the next logical question would be, what is your favorite ice cream then?
It depends on what I've eaten before, or if I only eat that. But if I have to select one, I would say it's pistache, rigorously out of Sicily.
Delicious.
That are very difficult to import in Switzerland, by the way.
I think by now a lot of people now would like to try the ice cream of your wife. Maybe you can give them a hint where they could enjoy.
If you Google Ivan Martin ice cream, maybe something will come out. So I don't have to give any further hint.
Okay, beautiful. Thanks. Is there any message you would like to leave, particularly regarding the future of regenerative medicine?
I would like to conclude with something that I've already mentioned, and that I especially would like to recommend to the younger scientists who still have a career in front of them to develop. And so not to consider tissue engineering the way that we unfortunately did 30 years ago, naively thinking that, if you combine cells into a scaffold and you apply some stimuli, you will generate a tissue that will work. But to go deep into the biology, to understand the fundamental cellular and molecular processes that need to accompany the regeneration in that particular setting of that particular indication and type of individual. Again, there is no possibility of controlling processes if we do not understand them. This is what I have inherited in my engineering studies, and we need to better understand biology to achieve reproducible regeneration.
Beautiful. Thanks a lot for your time today. It was a real pleasure to have you on here on the podcast.
Thank you, Norian. My pleasure. Looking forward to the next ATiO meeting.
We hope you've gained as much inspiration and knowledge from this conversation as we have. Remember, the journey of innovation and progress is an ongoing one, and the contributions of visionaries like our guests continue to shape a brighter future for us all. Stay tuned for more engaging interviews and discussions. In the meantime, check out our website at atioalliance.org for more information and content. Thank you for joining us.